In this section, we're going to see how you get energy from food. So let's take a slice of pizza, for example. In the pizza, you have the crust, which the crust is a lot of starch, which is carbohydrate. You also have the protein. Pepperoni and cheese both have protein in them. And you have fat. The cheese and the pepperoni both also have lipid or fat. So when you eat a pizza, you're getting carbohydrate, protein, and lipid. Your digestive system is going to break these nutrients down. Remember chapter 2, polymers and monomers, you're going to break them down. When you break down and digest carbohydrates, you end up with glucose. Glucose is blood sugar, and here we have a picture of glucose. Glucose is going to be used for energy. When you digest the proteins, think back to chapter 2, proteins are made of amino acid. So your digestive system will turn the proteins into amino acids. You'll have amino acids in your blood. Now you can use amino acids for energy, but we're not going to get into that. When you digest lipids, one of the main things you get is triglycerides. And here is a picture of a triglyceride. And you can also use it for energy. So fats and carbohydrates are the main things you use for energy. And if we look at the triglyceride and the glucose, what they both have is hydrogen. Notice all these H's, all these hydrogens here in the glucose, hydrogens. So hydrogen is the main thing that you're going to use to get energy. So you eat food, you digest it into your monomers. They go into your blood. Your blood takes them to your cells, and then your cells are going to use the hydrogen that is in them to make ATP. Remember, ATP is the energy currency of cells. Then ATP will provide the energy for cellular work. So let's look at ATP now. ATP stands for adenosine triphosphate. So we have an adenosine. We're just going to put a big A here to represent the adenosine. Tri is 3, so we have 3 phosphates attached to it. So P represents phosphate. The lines represent the bonds, so these are bonded together. So these 2 lines here represent the bonds that are holding the phosphates to the adenosine. This one that's all curly like a spring is a high energy bond. So if you break this bond, a lot of energy is released. When your cell has work it needs to do, it breaks this bond. And when it does, you get ADP. So if you break that bond, the phosphate is no longer attached. It's just over here by itself. And that energy that was in the bond is released. So that energy is released, and it does work for the cell. Now you have ADP and PI, just a phosphate by itself. Well, now you recycle. If you think of this like your cell phone, this is your phone after it's been plugged in, and your battery is at 100%. You are set, you are good to go. Then you use your phone. So now here is your phone, and you're getting that little red warning that says battery low. So this is your battery is dead. So what do you do at the end of the day when your phone's battery is dead? You don't just toss your phone in the trash. You plug it in and you recharge it. That's what your body does with the ADP. You're not just going to throw it away, you just recharge it. So all you do is reattach that P. You create a new high energy bond and you reattach it. It's just like plugging your phone in and recharging the battery. Hydrogen is your charger. That is what you use to do this. So you use hydrogen that you get from your food. Remember, it's in the glucose, it's in the triglyceride. So think of hydrogen as like your charging cord for your phone. That's what you use to recharge this ADP and P back up to ATP. So now let's start looking at how this happens. And if you notice in your book, in your binder, it says like round 1, and then in a few pages you have round 2 and round 3. This is a topic, it's a very complex topic. It's covered in every biology class just about, and it's just one of those things that really gets students down. So I've broken it down into little bits, and I take a simplified view of it. So you may have learned a lot more detail and complexity in other classes. I try to slim it down to just what you really need to know to understand what's going on here. So cellular respiration is the process of how you use hydrogen from your food to take that ADP and PI and convert them to ATP so that your cell can have energy to do its work. So you have a cell. Here's a cell. And you may know from other biology classes, mitochondria is the powerhouse of the cell. That's where ATP is made. So we're going to give our cell a mitochondria. And you have blood. Remember, all of this nutrients are being carried in your blood. So here is your blood. And we're going to focus on glucose. Glucose is the main thing you use to make ATP. And the chemical formula of glucose is C6H12O6. So we have blood and we have glucose in it. You also have oxygen. We're going to put 6 oxygens here. So you're eating food, you're getting glucose, you're breathing, you're getting oxygen. These two things go to your cell. So your cell is going to take in the glucose. We're going to have glucose right here. It's going to go in the cytosol next to your mitochondria. Now remember, the overall goal is you want those hydrogens. First thing we're going to do is break the glucose in half. And when you break it in half, you're going to get pyruvates. Those are then going to go into the mitochondria. You're going to keep breaking it down. You're trying to get at the hydrogen. So you're going to break it all the way down so that you end up with just 6 carbons, 6 oxygens, and your 12 hydrogens. It's all broken up. Then you can use the hydrogens to make ATP. So you're going to have in your cell ADP and PI. They're going to come into the mitochondria also. You will use the hydrogen to push them together. So it's like you have to take your phone into the mitochondria because that's where the charger cord is and you charge it back up. You're going to use the hydrogen, push those together so that then ATP is produced. Then you're done with these, with the carbon, hydrogen, and oxygen. Your cell has to have a constant new supply. It can't reuse the glucose. So once you're done with those, oxygen is like the garbage man. It comes in, it's the cleanup crew. The oxygen binds to the carbon and the oxygen and the hydrogen from the glucose. And when it does, it produces carbon dioxide and water, 6 of each. Those are the waste products then that come back to your blood. So that's kind of the basics, the big picture of it. And with your ATP here, it would go to a place in your cell where you have work to do. You would break off that 3rd phosphate, release the energy to do work, and you would be back to having ADP and PI. So it's this ongoing cycle. So let's look at this in a little more detail now. We're going to break this up into steps. So we're going to have step 1 here is glycolysis. Glycolysis occurs in the cytosol next to the mitochondria. And this is where you have your glucose. So we have the C6H12O6. And we're going to break it in half. When we break it in half, we pull 2 hydrogens out. So first we pull out 2 of those hydrogens. And when you pull out hydrogens, they can't just be on their own. They have to be put on a carrier. So we have a carrier molecule called NAD. Just think of it like a taxi cab. So we've got a little taxi cab here. That's NAD. NAD is a taxi cab. It's a carrier molecule. It comes along to pick up those 2 hydrogens we just pulled out. So then we get to NADH. So that just means now there's a little hydrogen riding around in that taxi cab. Then we break what's left in half. So if you take C6, break it in half, you get C3. We had H12, but we took 2 hydrogens out. So that leaves us H10, and half of 10 is 5. O6 split in half is O3. We're going to have 2 of these. These are the pyruvates. C6H5O3 is pyruvate. When you break the glucose in half, it does release a little bit of energy, and you have a net gain of 2 ATP. Now, it actually releases 4 ATP, but breaking it uses energy. You have to use 2 in order to break it and get 4. So your net gain is 2 ATP. That's glycolysis. Take a glucose, you pull 2 hydrogens out, put them on your carriers, break it in half, you get 2 pyruvates, 2 ATP. So at the start of glycolysis, you had a glucose. That is what you started with. At the end of glycolysis, you have 2 NADH, 2 ATP, 2 pyruvates. That's what you have at the end. The next 2 steps I'm going to lump together here. We're going to have formation of acetyl-CoA and Krebs cycle. One of the things with biology is there's a lot of different terms for the same thing. What happened is hundreds of years ago, somebody named something. Then in more modern times, somebody decided to change the name. But then you have some textbooks use the old name, some textbooks use the new name. And so we end up just with multiple names for the same thing. Another name for Krebs cycle is citric acid cycle. In this stage, the pyruvates enter the mitochondria. And you're going to finish breaking them down and extracting the hydrogen. Remember, the hydrogen is what you want. That's your charger. So we have C3H5O3. We have two of those. These are inside the mitochondria now. We're going to pull out all of the hydrogen. So 5 here, that's 10 hydrogens get pulled out. Well, remember, whenever you pull out hydrogens, they have to be on a carrier. So we need 10 more little taxi cabs to come along and pick them up. We're going to have 8 NAD and 2 FADH. Just think of these as like different taxi companies. Think about if you go to an airport or a train station or something and there's different types of taxi cabs. That's what this is. NAD and FADH are just two different types of taxi cabs. They come along to pick up those 10 hydrogens. So you get 8 NADH and 2 FADH2. All of your hydrogens are extracted now. So we have all of the hydrogens on these carriers. Remember, we have 2 up here in glycolysis, 2 NADH, another 8 NADH, and 2 FADH2. So in the next step, we're going to use those hydrogens to make the ATP. But right now, we have to deal with waste. The hydrogen is the part you want. We have these carbons and oxygens. We have 6 carbon atoms, right? 3 here, 3 here. We have 6 oxygen atoms, 3 here, 3 here. They are waste. Think about when you go to the store and you buy a new pair of shoes. They come in a shoe box, and there's the box and there's tissue paper in there with them. Well, which part is it you actually want? Is it the box, the tissue paper, or the shoes? You spent all that money to get the shoes. The box and the tissue paper are just the packaging that your shoes come in. It's the same idea here. Hydrogen is what you want. Hydrogen is the shoes. It's what your cell is after. The carbon and the oxygen are just the box and the tissue paper that the shoes come in. So when you get home, you take your new shoes out and you put them on your feet, and the box and the tissue paper you throw in the trash. Okay, the carbon and the oxygen are waste. But you have to be careful of what you put in your blood. You can only put molecules in your blood that are safe to put in your blood. So this is where that oxygen comes in. If we can add 6 more oxygen atoms to this, then we could have 6 carbons, 12 oxygens. Well, that would make 6 CO2 molecules, and CO2 is safe to put in your blood. So you take 3 oxygen molecules to give you those 6 more oxygen atoms. So this is O2. That means it's a molecule atom bonded to an oxygen atom bonded to an oxygen atom. So for each O2, you have 2 oxygen atoms. So if you just take 3 times 2, that's 6. You get 6 oxygen atoms. This is basically saying you have 3 pairs of oxygen atoms. Well, if you have 3 pairs of oxygen atoms, you have 6 oxygen atoms. So we get 6 CO2. And this then is what you put in your blood. Okay, so you bought your shoes, you came home, you took them out of the box, you threw the box and the tissue paper away. That's what's going on here. Your cell got its glucose, It broke it up, it got the hydrogen, the part it wants, and it has thrown the carbon and oxygen, which we're just packaging, away. And your blood is where cells throw stuff away to. Now we're ready to go to the next step. Our next step is electron transport. This also takes place inside the mitochondria. And you have, it's called the electron transport chain. It's kind of like a slide. I think of it as like you go to the park and there's a sliding board. That is the electron transport chain. Back over here where I said those ADP and PI go back into the mitochondria, when they do, they go to the electron transport chain. So right here, we're going to have ADP and that P by itself. This is where the carriers take the hydrogen. So we have our 2 NADH here, plus 8 NADH here. That gives us 10 NADH. That comes up here to the very top of the electron transport chain, and it drops off the hydrogen. Remember, they're like a taxi cab. They pick up the hydrogen, they take it somewhere, they drop it off. So now the hydrogen is here at the top of the slide. The hydrogen goes down the slide. As it does, it pushes these together. So you get ATP. And then you end up with your hydrogen at the bottom of the slide, and you got 3 ATPs. Notice you have 10 of them. So 10 times you're going to do this. So by the time you do that 10 times, so you have 10 hydrogens, each one makes 3 ATP. You're going to get 30 ATPs made from those NADH dropping off their hydrogen. Okay, we also have our FADH2. It's the other brand of taxi cab. It drops its hydrogen off right here. Now notice it's not at the very top. It's kind of part way up. So this hydrogen, when it goes down, it makes 2 ATP. So we have 2 of these, and they each make 2 ATP. So that gives us 4 ATP. So the FADH misses that top one. It starts part way up the slide, and it makes 2 ATPs for each hydrogen it drops off. So this means altogether, when all 12 hydrogens go down that slide, you get 34 ATP. So electron transport produces 34 ATP for you. Now you have 12 hydrogens at the bottom of the slide. Remember we said hydrogen is like shoes. You buy a new pair of shoes, you wear them around for a while. Well, eventually they start to wear out. They get old and smelly, right? Like you're not wearing the same shoes now that you had 10 years ago. You buy new shoes now and then. Well, these hydrogens, by the time they get to the bottom of the slide, they're all old and smelly and worn out. It's time to get rid of them. So these are now waste. You've made your ATP, you're done with them. So again, you take in oxygen. Notice back here, I put 6 oxygens in the blood. We used 3 of them here. Well, this is where the other 3 come in. If you have 12 hydrogens, and remember 3 O2s, that's 3 pairs of oxygen, so that's 6 oxygen atoms, you can make 6 molecules of water. And this is called metabolic water. It's metabolic because it is made by your cells. So whenever you're doing cellular respiration, your cells are actually making water and putting in your blood. So this goes to your blood. So that is why over here we had 6 carbon dioxides and 6 waters going back to the blood from the mitochondria. So now let's summarize this. Let's put it all together, the whole big picture. You started off with C6H12O6. You also had 6 oxygens in the blood. We end up with 6 carbon dioxide and 6 water. Right? We're just looking at what was originally in the blood that leaves the blood and goes to the cell, and then what comes back to the blood. Now what goes in has to equal what goes out. So let's check our math and make sure we did this right. We're going to have carbon, hydrogen, and oxygen atoms. On the left, so this is what leaves the blood and enters the cell. And on the right is what leaves the cell and goes into the blood. We need to make sure these two sides equal each other. So carbon, here we have C6. So that is 6 carbons. Here we have H12. That is 12 hydrogens. Here we have 6 oxygens and another 6 pairs of oxygen. So this is 6. 6 pairs is 12. 6 and 12 is 18. So we have 18 oxygen atoms. Over here, 6 CO2. It's going to be 6 carbons. Here we have 6 waters. Well, each water has 2 hydrogens. So if you take 6 times 2, that's 12. 6 CO2, there's 2 oxygens in each of those, and we have 6 of them. So that's 12 oxygens there. 6 waters. Each water has 1 oxygen, and we have 6 waters, so that's 6. 6 and 12 is 18. It all adds up. So all you're doing is converting glucose and oxygen into carbon dioxide and water. The reason you do this conversion is your payment. What does your cell gain by doing this conversion? Well, it gets 36 ATP. We had 34 ATP here at electron transport. We had 2 ATP here from glycolysis. So that gives us 36 ATP altogether. You also get heat. If you look at page 73 in your book, it talks about the second law of thermodynamics. Whenever you convert energy from one form to another, you get heat. We were converting energy. Glucose is a form of energy. ATP is a form of energy. We converted glucose energy to ATP energy. Therefore, we also produce heat. So this is where energy comes from. This is where body heat comes from. So you can kind of think of this as your cell's job is to convert glucose and oxygen to carbon dioxide and water. And just like how you get paid money for doing your job, your cell is paid 36 ATP and heat for doing this job. So think about this. Let's put some implications to this. You get hot when you exercise and you shiver when you're cold. Well, whenever you use ATP, in order to make ATP, one of the payments you get is heat. So the more ATP you make, such as when you exercise, the more heat you produce. So when you exercise, you need more ATP. Therefore, you make more heat. And this is why you sweat. You sweat to get rid of that extra heat. Because remember, homeostasis, you want to stay at 98 degrees. Now, if you are cold, you will shiver. Shiver means your muscles are working. If they're shivering, they use more ATP. And if you use more ATP, you're going to make more heat. And this will warm your body up to homeostasis.